Have you ever just thought, what type of genetic mutations are there? Oh, no, just me? I guess I'm just a little weird. Hello people of the internet and biology class, my name is Stickman and I have been called upon to help explain mutations. So let's start. To start, we need to talk about just what a mutation actually is. A mutation is a change in the nucleotide base sequence of a gene or DNA molecule. So now that we know what a mutation actually is, you guys are probably wondering what are the types and how many different types are there? Well, to answer that, there are two types of mutations. One is called chromosomal mutations, which affects the chromosome structure by gain, loss, or rearranging. And the other one is point mutation, which affects a single nucleotide by loss, gain, or substitution. Let's start off with the easy one, chromosomal mutations. There are four major types of chromosomal mutations. The first one is deletion, which is the loss of a piece of a chromosome. The second one is inversion, which is when a segment of a chromosome breaks off and flips. The third one is translocation, which is where a piece of a chromosome attaches to a non-homozygous chromosome. Now I know what you're thinking. What is a non-homozygous chromosome? Well, non-homozygous chromosomes are chromosomes that are not genetically similar. Take, for example, the chromosomes that are on screen right now. The one to the left is type A, B, C, D, E. While the one on the right is E, F, G, H, I. So they're not genetically similar because they don't share the same type, meaning that they are both non-homozygous chromosomes. Now back to chromosomal mutations. The fourth one and the final one is non-disjunction, which is when a chromosome fails to separate from its homologue during meiosis. Now that we know most about chromosomal mutations, I think it's about time we start talking about point mutations. But we first have to talk about RNA, which is how these mutations can actually happen and what can affect it. So first, DNA is found inside the nucleus of the cell. We all should know that. It's basic genetics. We also should know that it stores all the information about the cell and for the cell. In order for that information to go out into the cell and be used, DNA must first become RNA. In order for DNA to become RNA, the DNA must split. As you can see, there are four nucleotide base pairs that make up the interior. It goes T, A, C, G. T and A go together, just like how C and G go together. When it splits, TACG combines with floating nucleotides around inside the nucleus. T goes to A, C goes to G, and G goes to C. Now, I know what you're probably thinking. Well, A goes to T, of course, because that's how it is in DNA. But that's not actually what's happening. There are no T nucleotides inside the nucleus, but there is a U instead, which is what will bond to the A. Now, I know that was a lot to take in, so let's do a recap. DNA cannot leave the nucleus, so it has to split, creating RNA. Whenever it splits, it has to pick up nucleotides. T goes to A goes to U, C goes to G, and G goes to C. Now that the information can leave the cell, it would go to the ribosomes, which are the protein makers. I shouldn't have to tell you just how important protein is to us. So if it gets made wrong, there can be serious consequences. When the RNA reaches the ribosomes, they will start reading the RNA. The ribosomes read each grouping of 3 nucleotides, which corresponds to a specific amino acid. These are called codons. The codons make up a chain and the ribosome then reads that chain and makes proteins based off that information. There are codons that tell the chain when to start and stop. Now, let's start with point mutations. There are 3 types of point mutations. The first one is substitution, where one nucleotide replaces another. This can cause the codon to change and tell the ribosomes to make the wrong amino acid, or it may say the same thing as the other one did. For example, let's say that the codon is CCU. Its amino acid is PRO. Now let's say that the U changed to a G. The amino acid wouldn't change, but if the first C were changed to a U, it would become a different amino acid, named SCR. Now for the final 2. The first one is deletion, where one or more of the nucleotides in a gene are lost. The last one is insertion. This is where one or more nucleotides are added to a gene. Both of these guys can cause frame shifts. You guys are probably thinking, what in the world is a frame shift? A frame shift is caused by either deletion or insertion. They force all the amino acids downstream to change. Think of it as if you were restricting the flow from a river. From that point on, the river is changed. It is important to know that substitution cannot cause a frame shift because it only changes. Well, that's all I have for you guys today.